is indeed a pleasure to welcome you back as we embark on yet another day filled with engaging discussions on groundbreaking research and insightful sessions. As we move forward, I am delighted to welcome you all to the scientific sessions of AdtU Pharmacon 3.0, where we continue our journey of exploring groundbreaking advancements in pharmaceutical sciences. This session, like the others, aims to contribute to our overarching theme, empowering healthcare and well-being towards achieving sustainable development goals, research, innovation, and industry-academia partnership. Today's session promises to bring valuable insights and expertise from one of the leading minds in public health and healthcare systems. Now, it is our great pleasure to introduce our esteemed guest, Dr. Supratim Vishwas, Regional Project Analyst, HSS UNDP Guwahati, a renowned public health expert with an impressive 18-year journey in the field. As the current leader of Health Systems Strengthening Team for UNDP in the Northeast India, Dr. Supratim has been instrumental in driving transformative changes in public health landscape. With a distinguished career spanning multiple states, including Rajasthan, Tripura, and Assam, Dr. Supratim has made significant contributions to various initiatives such as the rollout of digital health innovations, COVID-19 response efforts, and projects focused on water sanitation, hygiene, and social health activism. A published author and a lifetime member of the Indian Public Health Association, Dr. Supratim's expertise and passion for public health have made a lasting impact on the lives of countless individuals. Please join us in extending a warm welcome to Dr. Supratim as we look forward to gaining insights from his vast experience and expertise in the field of public health. Thank you, sir. Sir, you are invited to the stage. Dr. Vishwas.
चाहिए क्या ये लिखिए तो ज़्यादा हो जाएगा हाँ ठीक है ओके थैंक यू Good morning, all of you. Good morning. Okay. So, thank you for inviting me in such a good platform. So, I must convey my gratitude and sincere thanks to Dr. Saikal Sen, who is the HOD and Dean for your pharmaceutical department of the Eastern University, Assam Downtown University. I would like to convey my sincere gratitude to all the faculties who have coordinated for this session with me since last 1 or 2 months. And also, I would like to convey gratitude to all the guests present over here, the students, and the other officials who are associated with this program. So, it's my pleasure to be here. And already I must thank to the organizer to introduce me. And I was just, when I was coming to the university, I was seeing the posters and displayed with our photos and all those things. So many thanks to all of the organizer for this. So coming to this technical session, the topic I have chosen to discuss with you all, this is a digital innovation meets public health experience from India. So let me tell you, I'm working for United Nations. So I'm the regional lead for the United Nations. Currently, I'm in the role of regional lead for health system strengthening. But earlier, I was posted in Tripura and Rajasthan, Jaipur, as a state lead for there. So for last 6 years, I'm working for the United Nations, and we have some digital innovations that we are supporting to the Ministry of Health and Family Welfare, Government of India, as well as our presence is there in all the 34 state and UTs in the country. So UNDP is a UN agency, all of you are aware, United Nations Development Program, which are currently having few digital innovations, digital tools. By that, we are supporting to all the state governments, district authorities and all. So coming to my slides, I would like to, this is not related with the pharmaceutical thing, but this is, this is related with the public health and how digital tools are changing our lives, how it is supporting to change the public health scenario in the country, even in the remote areas, in the rural areas. That I want to give details on that. I will take you in this journey, how we have started this, what is our scenario right now, and how we will look forward in coming days. So the topic of this conference is Sustainable Development Goal and Digital Innovation. So here I want to just focus on how SDG, the Digital Acceleration Agenda, is supporting, the digital innovation is supporting the Sustainable Development Goals. So you can see the United Nations through initiatives like the SDG Digital Acceleration, recognition the crucial role of digital technology in achieving the SDGs. So what UN think, this 70% of SDG targets can be fulfilled by the digital innovations and digital tools. And digital technologies can play as a catalyst for improving the public health scenario in the country. If we are talking about the SDG digital initiative, we can see the specific areas of impact. What I was telling to you that digital innovations can accelerate the SDG goals. So here I only mentioned 3 SDG goals. There are so many SDG goals are there, targets are there, and digital innovation obviously can accelerate all the SDG targets, but relevant to my topic, I want to highlight only 3 SDG goals. This is the education, health, and partnership for the goals. This is the SDG 4, SDG 3, and SDG 7. So if you look into the 3 SDG goals, this is the health is the SDG 3, and the education is the SDG 4, and partnership for the goals, SDG 7. In both the SDG goals, the digital technologies can bridge the educational gaps, can improve the healthcare access, and also can play a crucial role for public-private partnership from bringing digital technologies to all the nations, peoples, and communities. My content in this topic is I will cover 3 digital technology. That is, one is the EVIN, one is the COIN, and number 3 is the UIN. Anyone know about this 3 digital tool? Anyone can, because talking only one side is not that much fruitful. Anyone can recall any of the 3 digital tool? This might be new for you, but second one is not the new, because we have already vaccinated in the COVID vaccination, and that was the application COVIN. But other 2, the EVIN and UIN, that might be new for you, but this COIN is we are all familiar with, because we have been vaccinated in COVID vaccination through this platform COVIN. Okay, so UNDP, United Nations, is supporting these 3 digital tools. The COIN, the COVID vaccination, you can recall, taking the slot, appointment, we are not getting the appointments, so these things, So that initiative was supported by us. UNDP supported the ministry or all state governments. That access was with us. How we can give the slot, booking, and everything. So these are the three digital tools we have utilized. We have been successful in India. So coming to the first EVIN, I want to show you. This is the basic tenets of digital health system. This is the road, this is the journey. What UNDP, United Nations, is supporting the currently in Ministry of Health and Family Welfare, Government of India. So EVIN is a digital tool which is ensuring the vaccine stock and management. You know all the children must be immunized. The pregnant women, after declaring the pregnancy, she must be given 2 doses of tetanus, TD, and the children, the infant and children, they should be immunized. So the vaccines, how the vaccines are supplying to the health facility, this is the digital tool is there to maintain the accuracy of the vaccines. The EVIN, you can see the last one. The EVIN was introduced in 2015 by Ministry of Health and Family Welfare, Government of India, and the technical partner is the UNDP, United Nations. After that, in 2021, you can see then the road, COVID was there. COVID was launched for COVID vaccination. In 2024, we have, after the successful rollout of EVIN and COIN, now we are implementing UIN. I'll give you details what is the UIN and all. Then there is another portal, that is the TB UIN for weaning over TB tuberculosis. So there is a adult vaccination program for above 18 years age group, and that is also captured in the TB UIN portal. And last one is the AVIN and JUIN. Right now, currently, UNDP is supporting from Minister of Health for the human vaccines, human health. But right now, means from now the process already started to work for the animal health also. So now we are working with the animal husbandry for ensuring the vaccine efficacy for the animals. And for that, we are introducing 2 portals. One is the AVIN and one is the JUIN. So this is the digital pillars, what I told you. The first digital health solution was the EVIN in 2015. After EVIN, when COVID pandemic was there, then there was a crisis. All of you are aware that there is a need for giving COVID vaccination, and all the COVID vaccination need to be captured in one platform. And that time UNDP came forward to introduce this COVID platform, and through COVID platform we all are being vaccinated. Till now we have the digital certificate that is required for international travel and everything. Number 3 pillar is the UIN. So after successful implementation of EVIN and COVIN, now we are implementing UIN. This is a digital platform for routine immunization for children and pregnant women. Now the pillar 1, the EVIN. I was telling from my first slide, there is a EVIN, Electronic Vaccine Intelligence Network and all. So EVIN is basically, the full form of EVIN is Electronic Vaccine Intelligence Network. What is that EVIN? How we are supporting Government of India, and how the people, community has been benefited from this EVIN. Let us see. So there are 3 critical questions for program managers. What are the 3 critical questions? 3 critical questions is, where are my vaccines? Are they stored at recommended temperature? Are they stored in adequate quantities? These are 3 questions. And let me tell you, this is not a digital solution. This is actually saving lives of our children. And you must be, here you people all are gathered, and I know most of the people who have children at home, even before joining in the United Nations, I also went to a private practitioner to vaccinate my child. Because we have a perception, na, ki my child will be vaccinated. Why I'll go to the government facility? There nothing is good. We have to go to the private facilities, private practitioner, the good big hospitals, private hospitals. But why we go to the private hospitals where the vaccines are free at the government? Because we have a pre-mindset that in government, the vaccines are not good. In government, there will be very queue, line will be there. And in private, I will go, I will pay, and my child will be vaccinated. There are another theory, painless vaccine. There will be no pain, and my child will be not suffering from fever and all. Let me tell you, all the vaccines, the fever and all those things, that is the symptoms, and that means if fever comes, that means your vaccines is working. I'm not going in what is the painless vaccines are not good and not. I'm not going into that. That is another discussion. But as per the government of India, every vaccine has some effect on the body, and that is fever comes, it means it is working. It's the number one. Number two, when we are going to the private practitioner, our child has been vaccinated. But have you seen any time where the private practitioner are storing their vaccines in a normal freeze? If there is a load shedding, if there is a power cut for more than 8 hours, more than 6 hours, they will shift the vaccines to another freeze? I don't think so. I'm not talking about the Guwahati. Please think about other northeast state also. I am from Agartala or some other states, Arunachal, where the private practitioner will, if power cut is there is no system. But yes, in government facility, the vaccines are stored with recommended temperature. If in a government facility, vaccines are stored and there is a power cut more than 2 or 3 hours, there will be alarm. We have, under this EVIN program, we have installed the temperature logger. If you visit any of the health facility, you can see there is a device is there. I will show you in my slide. That is the temperature monitoring device. If there is a power cut or if there is any fault in the cold chain equipment where the vaccines are stored, the alarm will be there, and the medical officer of that health facility will receive a message, SMS, your vaccines are crossing above the recommended temperature. So today, as on today, the vaccines are very safe at all the health facilities because we are monitoring digital. Even right now I can show you from this mobile the temperature of any health facility in any of the district in Arunachal, in Assam, in Tripura, in Manipur. From here I can see what is the recommended temperature. So that is not possible in the private sector. So this is the benefit of this digital solution. So why EVIN was introduced? Because there was a challenge, difficulty inputting data online at district level, right? Availability of functional computer, internet access, availability of data entry operator, and electricity supply. These 4 things are the challenges. So what we have done, we have introduced a EVIN solution. There is 2 things. In EVIN, Electronic Vaccination Network, there is a 2 things. One is one like this digital application where whenever the vaccines are going from the state vaccine store to district, even at the health facility, there will be all the cold chain handlers will enter the data. So we can see from my mobile how many vaccines, how many BCG, how many Penta, how many hepatitis B vaccines are available at a health facility, government health facility. Okay. After that, we can see this is a temperature logger. You can see the right. So this temperature logger has been installed all the cold chain equipment, that is ILR. We call ILR. In that ILR, this temperature logger has been installed. So with these smartphone and temperature loggers, we can access the availability of vaccines, temperature of vaccines, right away from our Android phone. This is the temperature monitor. You can see when at any time, if there is a temperature breach for power cut or for fault in a cold chain equipment, there will be alarm from this device. Okay. So this is all about the electronic vaccine intelligence network that is implemented in all the government health facilities. And please pass this message कि vaccines are now very safe at government health facility rather than private health facility, means private chamber, because in government health facility we have these mechanisms and your child can be safely vaccinated at any government health facility. Now coming to the second pillar, that is the UIN. UIN is a universal immunization program. That is, you can recall during the COVID vaccination, you have a COVID vaccination certificate where your first dose, second dose, precaution dose, everything has been registered. You have certificate, no? During the COVID time, all of you have certificate. For the routine immunization, my child, your child, or the pregnant women who have been vaccinated, they will get a vaccination certificate like COVID vaccination. So, routine immunization program has been digitalized through this new platform that is called UIN. So, the UIN was launched by Honorable Prime Minister in October 29, 2024, just 6 months back. So, Honorable Prime Minister has introduced that UIN in the Pan-India. Now, in Pan-India, this UIN platform is there to support the routine immunization program. Why the rationale for developing UIN? Why UIN has been developed? What is the necessity to bring another portal in place? Because manually every healthcare workers, they are maintaining the data from routine immunization. But what is the rationale for developing UIN? Because from this portal, you can vaccinate your child from anywhere. Like you know that your child will be vaccinated in 1.5 months, then 3 months, 6 months, 14 weeks, like that. So if in a 1.5 month your child is vaccinated in Guwahati and now he or she is going to Mumbai, so from that mobile number, there is a unique identity number, is mobile number. From that mobile number, he or she can be vaccinated in Mumbai in any health facility, and the digital certificate will be generated from this portal. What are the challenges? Why UIN is supporting the routine immunization? Because the challenges is that we have multiple vaccines and multiple doses, leading in difficulty by monitoring. Immunization at private health facilities not recorded. Mobility of beneficiaries, beneficiary awareness. So these are the challenges. And now in UIN portal, we have provision of live report, national level still session site, inclusion of private sector, and you will be happy to know, till now in India, UIN, this portal has been rolled out in only government health facilities in total pan-India. But just from this month, March, UIN has been implemented in private health facilities, and Assam has been taken as the pilot study in Guwahati. In 5 hospitals we are doing the pilot study. So Assam is the first state in country where the private hospitals, this portal will be rolled out. Now we have started from the largest private hospital where vaccinations are done in a larger quantity, like Pratiksha Hospital, other some 2-3 hospital, Apollo, and I think charitable healthcare, some Catholic hospital is also there in Guwahati. So in those private hospitals, we have started at a private pilot basis, that UIN and the private hospitals. So the UIN, this portal has been rolled out in scale up from January 23, and national workshop was held in 8th and 9th June 2023, and now it has been inaugurated by Honorable Prime Minister. So the immunization services, we have digitized the total immunization services. There are few modules you can see in the right corner: the administrator module, delivery point module, vaccinator module, mobilizer module, and registration scheduling module. So in that module, like one baby is born in the hospital, he or she will be given the birth doses after the delivery. Within 24 hours, we have to give the hepatitis B. If child is missed from 24 hours, so he and she cannot be given hepatitis B in a lifetime, because hepatitis B has to be administered within 24 hours. So this hepatitis B, BCG and OPV 0 dose, that can be given at the birth, at the labor room, and through this UIN portal, these will be recorded. After delivery, when the baby will go to the home, and after that he or she will be scheduled for so many vaccines. So all those vaccines can be given at the government or private health facility and through this UIN portal. And another benefit of this portal is beneficiary can see, means from mobile you can see for tomorrow which health facility is nearby to you and where the sessions are going on. So you can book online appointment from your mobile phone for any government health facility or any private health facility for this vaccination. So no need to go and check in the health facility. You can see in the mobile phone whether tomorrow's session is there or not. And UIN portal has been integrated with several other portals, like must be aware about the ABDM, Ayushman Bharat Digital Mission. There are few other digital technologies that there, like health professional registry, health facility registry. So all those ABHA ID, now all has to have ABHA ID. So those ABHA IDs and other unique IDs can be created through this UIN portal. So there are integration in between UIN and Ayushman Bharat Digital Mission. So the benefits of UIN for citizen. What is the benefit? I am talking about the UIN and all. You must be thinking, what is the benefit for the citizen? So benefit for the citizen is first one is the online self-registration. You can register yourself. You can take appointment also. You can generate your ABHA ID from this UIN portal. You can search vaccination centers across the country. You can, I already told you can schedule appointment. You can digitize vaccination record. You will get a vaccination certificate after completion of your vaccination. Okay. SMS notification will be given to you. Like after 1.5 months, when the next vaccination is due, in 6 weeks, 9 weeks, 14 weeks, all the SMS will go to your registered mobile number. Your baby is due for these vaccines. Please come on this health center on that day for your vaccination. So these SMS and reminders will go to the beneficiaries. Ensuring dose intervals, provision to get vaccinated across the country, anywhere you can get it. Provision of report side effects of vaccinations. If there is a side effect, means fever is not a side effect, it's a minor side effect, because fever is normal for any vaccination. But apart from fever, if any minor or severe side effects is there after your vaccination, you can report. That is called AEFI, adverse following vaccination. So adverse event following vaccination, AEFI. So that can be recorded, that can be reported in this portal. So these are the benefits of UIN for citizen. So this is the certificate what we are talking about. This is the certificate will be generated after vaccination. So here you can see all the vaccines have been given, and with due date that will be registered. You must be knowing that whenever our child need to go to the foreign for study and everything, then this vaccination certificate is required, and we have to search from our file this MCP card or this certificate is there or not. But nowadays you can have this digital certificate on your mobile phone, in your DigiLocker, and from there you can get your vaccination certificate very easily. So learning from 10 years journey of building health systems. So this is a 10 years journey, because in 2015 the EVIN was launched, EVIN, UIN and COIN. Now we have already crossed the 10 years. So if we can divide our learnings in two parts, one is the governance, one is the technical. So in governance, what is the learning? We have a strong government ownership. I mentioned in my beginning, in all 34 states and UT, we have a strong government ownership. Then dedicated and skilled human resources are there for this program. Building standard operating procedures. We have a standard SOP. Regular capacity building and review mechanism is there for uh, uh, making the healthcare workers updated and giving them updated information on the portal and on the routine immunization updates. So these are the governance part. If we come to the technical part, number one is the traditional database to big data architecture. Now we have a big database of more than in Assam, if we see, more than lakhs of children and pregnant women have been vaccinated, and more than lakhs of vaccinations are recorded in the UN portal. Number two, the authorized access to the security and audit compliance, licensed product to sustainable open source solution, and obviously data triangulation to API based interportability. So now, based on this artificial intelligence, based on the digital tools, and we are being able to make partnership with the government. Government is also taking forward these portals, this digital solution till the rural areas, in the community level. Even ASHA workers, you must be aware the ASHA workers are also now using these portals to track how many beneficiaries are due in her area. So ASHA workers we have all ASHA workers in Assam are already registered in the portal, and they can download the due list. Even few ASHA workers, if they are not able to download from the mobile, our healthcare workers support them to download those due list. And with that due list, they go to the community, they ask the community, please come to the immunization centers for immunization, because there are some barriers also, some resistance, some reluctance also in the community, in few community. If we say in the tribal areas, in the minority areas, some areas we have some reluctance, barriers. So with these ASHA workers advocacy, we can overcome those barriers also. So these are the 10 years journey of the governance and technical part. So these are all about how the digital health solution is supporting all the 17 SDG targets in India. So these are the experience I'm sharing with you all. If you have any question, any queries, feel free to ask, please. If you have any questions, please feel free to ask. If no queries, then thank you so much. Thank you for listening me, and thank you for your patience also. Thanks. Thank you all.
Thank you so much, sir, for your enlightening and insightful presentation. Your work and expertise have provided us with a deeper understanding of healthcare systems and sustainable health solutions. Now I request Dr. Niladri Ghosh, Professor Faculty of Pharmaceutical Science, AdtU, to felicitate Dr. Vishwas. As we conclude this session, I would like to express my sincere gratitude to Dr. Supratim Vishwas for sharing his knowledge and expertise with us today. The next session will start very shortly. Thank you. You, you are requested to not leave the campus. You are requested to not leave the auditorium. The next session will start shortly. Kindly please wait. To chair this session, we are honored to have Dr. Mahendra Kumar Modi with us. Dr. Mahendra Kumar Modi, Professor Executive Dean at Assam Downtown University, is a distinguished academic and researcher specializing in bioinformatics and genomics assisted breeding with focus on improving rice varieties for abiotic stress tolerance. His prolific career includes over 50 peer-reviewed publications in renowned journals addressing topics such as GWAS for blast resistance, drought-responsive QTL mapping, and transcriptome profiling in various plant species. We are privileged to have you with us, sir.
Ladies and gentlemen, I, AdtU, your host for the next scientific session, welcomes all of you to the third offline scientific session of AdtU Pharmacon 3.0. It is now my privilege to introduce our distinguished speaker for the next scientific session, Dr. Biman B. Mandal, Professor and Associate Dean Academics, IIT Guwahati. Dr. Biman B. Mandal is a professor at Department of Biosciences and Bioengineering, IIT Guwahati. His present research is focused on studying natural biopolymers in the particular use of silk as a biomaterial for tissue engineering and regenerative medicine applications. He holds a PhD from the Indian Institute of Technology, Kharagpur, and has a postdoctoral from Tufts University, Boston, United States of America. Professor Mandal has published more than 210 plus research articles with over 13,750 plus citations and has an H-index of 66. He has 23 patents and licensed 5 healthcare technologies to industry with 1 product in market. Professor Mandal has received numerous national and international awards for his research contribution, and the most prominent ones are the Swarna Jayanti Fellowship, B.M. Birla Science Prize, Ramachandran Natural Bioscience Award, INSA and NASI Young Scientist Medalist, and the 3D Printing World Award. I now invite Dr. Biman B. Mandal, sir, as we look forward to gain insights from his expertise and research experience in the field of biomaterials and tissue engineering.
For master students.
Everyone. Sir, please join the speech.
A very good afternoon. Thank you for being here, and I know I'm standing in between you and your lunch, but trust me, the next 40 minutes or so, what I've been allocated, I'll try to take you through a very interesting topic, which probably you can see here. And this topic is something which probably pertains to all of us, which is about healthcare, right? So, do I have a switcher or something? Okay, fine, leave it. So, yeah, so as was introduced, I come from IIT Guwahati, which is one of the IITs in the state of Assam. And how many of you have visited IIT Guwahati? Anyone? Okay, some hands going up. If you haven't, I think you should visit, and we have a nice campus like yours here, and these are some of the pictures of our campus, which we are very proud of in terms of the beauty. And in this scenic campus, we have a lot of department school centers where we do a lot of research on, and my topic covers something which is on the health engineering, which is about bioengineering and how we can create tissues. Now, we all are fascinated by the fact that when a lizard or a tadpole loses its organ or a part of the body, it regrows it back. We have also have read about Harry Potter, and we are fascinated by the fact that the phoenix can rise from the ashes once it is burned. So the concept is, can we in human beings regenerate ourselves or create our part of the body in the laboratory? Think a day when it will come, like you are ordering your daily use items from the Amazon, the patients can order organs and tissues from the similar platform, and it will be delivered to the hospital before a surgery is performed. This is something, which looks like here's like a sci-fi movie, but this is what the future is all about. And I know we are proud and we are privileged to be working in this domain where we are trying to cater to some of the organs and tissues. So, why there is a need? First question. So there is a need because because of unfortunate traumas or disease that we intervene in our life. We are dependent on donors to get an organ or some sort of replacements. Now we also understand that the number of donors are very, very few compared to the people who are requiring these substitutes. So what would be a solution is something, if you can create them in the laboratory right? Make them similar to what we have in the body so that it can be transplanted. Sounds easy, but it's not so easy, and that's what the science we try to do of creating these organs and tissues in the laboratory so that it can be transplanted to the humans for saving life. Now comes the picture. Okay, before I go to what we do, I'll give you some glimpses. This is a quick snapshot of the work that we do in the laboratory, and probably you can see a lot of different kind of tissues and organs we work on. We work on trying to build bones, we work on liver for transplantation, we're working on kidneys, we're working on pancreas, we're working on skin, we're trying to heal wounds, so many things, right, we're trying to do. And the whole sole intention is to develop these technologies so that there is a solution for somebody waiting in the hospital bed for a new life. Now, how many of you are aware of the topic of tissue engineering? Any one of you? Not many people, okay. Why I'm asking you is because accordingly I'll tone down, and not go into the technical jargons. I'll try to make it as easy to understand so that, maybe you can get inspiration from this, and take up this topic for your future studies, right? So tissue engineering is something very fascinating where what we do is we take cells from the body. If you look at ourselves, we are made out of different kind of tissues and organs, and these tissues and organs are made out of cells and the polymer and the material they secrete, which we call the extracellular matrix, and together they form the tissues, the organ system that we are made of. So what we require to make them is cells. We grow them in a cell culture condition, then we grow them in a template which is a scaffold. What is a scaffold? Because the cells cannot form three-dimensional organs. For example, if you take a liver cell, you put it and you grow them, you cannot form three-dimensional liver. It will never form. It will form two-dimensional sheets. How do you form three-dimensional structures? You have to give a template, which is a three-dimensional form. You grow your liver cells, give the right set of conditions, then one can do. That's the easy part. Now, if you look at cells, probably we do not much appreciate we are built out of. So if you look ourselves, what we are built of, if you look at any organ, any tissue in the body, it's a very fascinating thing what we are built of, right? It's everything works together so as to, give a meaningful purpose to what we are doing. If you look at any kind of cells, skin cells, liver cells, kidney cells, bone cells, they are all adherent, but they are all mobile, they are all moving. Like you and me, they have their nutritional requirement, meaning they require to be given nutrition, food, so that they can survive, they can divide and proliferate and do the physiological activity in an organ or a tissue. And what is very interesting is, if you want to grow them in a scaffold, the scaffold is nothing but a sponge-like material which allows these cells to grow because of its large surface area, and they will grow and proliferate. And that is what we have to build. Now, one thing what is required for tissue engineering or engineering these organs outside the body is understanding the shear forces. What does shear forces do? These forces of compression, tensile, torsion, which we feel in our daily life while we do our regular activity, our exercise, allows these organs to mature and become fully functional. For example, if you don't do exercise, your muscles will be lean and thin. You will not have a bulging body, because if you put stress, take those loads, then the muscles would proliferate by taking additional nutrition. Same thing, if you do not walk or do not do certain activities, your bone would demineralize, and you will not be as strong as now. Similarly, the cardiac muscles also, you have to do a cardio so as to keep it in healthy situation. So for maturity, all our body doesn't keep anything for extra. It only provides you with that function when it is required, and the requirement is understood by virtue of the forces and the other forces they feel in the body. So having an understanding of the cells, the materials, the scaffolds, and the physical forces, we try to understand how we can, so if I say, okay, I want to create a liver or a kidney or a blood vessel or a cornea or an eye or skin, how I have to approach? The approach is what people are tweaking globally is form follows function, meaning if I have to have a function, I have to replicate the form both externally and internally. Internally means down to the level of micron and sometimes nano, so as to achieve it. Now, one very interesting thing we do, which probably would be very fascinating to you being part of Northeast, because we're blessed to have some very interesting materials, which is silk. Silk, we are very, Assam particularly is very popular, right? We have Muga silk, then we have Tassa silk in Bengal, we have Eri silk also this side, and these are all wild silk. Now silk is very popular because of the textile material that we do, that we wear, whether it's the sari or any kind of dress apparel. We use the same silk, which is a protein, to make human organs. Now you must be thinking, what silk has to do to making a human organ? Now the relationship is such that silk, as I said, is a protein. Protein are made out of amino acids, and it is the same amino acids that our body is made out of, right? So the whole concept is using silk, making into different kind of scaffolds and growing cells on them. Of course, they have to be compliant because any material you put inside the body, it has to be not rejected by the body. So it has to be immune compliant. So the material has to justify and qualify some properties and regulatory approved. So silk, for example, Bombyx mori is already FDA approved, and we are working for taking Muga silk and Eri silk for those kind of approvals so that it can be put into human application. Muga silk, Bombyx mori silk is already in human applications in Europe and American market. Now to build an organ, we have to depend on different kind of techniques. There are many kinds of techniques, and we use all of them depending on what we are creating. I'll not go into the details, but one very interesting technique that we use now in today's time is 3D printing or 3D bioprinting. You may have heard about this term. So this is an automated version of printing where you give instruction, it feed the design in a machine, which is a 3D printer, right? And it makes an exact copy of what the design you have put in. So people make toys, people make different kind of utensils using polymers, plastic and all. Now, can you imagine we use similar kind of printers which are called bioprinters, more advanced version, and we can create human organs. And we do create in the laboratory in less than 30 minutes or 15 minutes. It's like imagine a patient going to the clinic, a hospital where a bone is traumatized. It's beyond repair by conventional means. What they would do, doctors would take an MRI scan, they will get the size dimension, put it into this machine. This machine will use a software and make it 1 micron thin sections, right? This will feed into this 3D bioprinter. This bioprinter would have ink or polymer. Then we put the patient's own cells or donated cells from other or individuals into that, and it would print that organ in less than an hour depending on the volume what we are printing, and that's exactly what we do in the laboratory. So this is very, very exciting thing. As I was saying, it creates replicas of human body parts in the laboratory, and that this technique is very, very powerful, right? And this printing is called bioprinting because it prints not only polymers, it prints biological cells which are living cells. Now printing a polymer probably is easy because it is non-living, but cells which are living, right, which is very sensitive to temperature, nutrition, culture conditions, sterility, it should not be contaminated, is a difficult proposition, but this has been mastered. These machines are available. They are quite expensive, but it is doable. Now this polymers, the ink that we use called a bio ink, which is very, very special to the kind of tissues that we print. And as a lab, we have developed a lot of these bioinks, and our motto is to translate many of these technology I'm going to take you through to the market as products. Some already we have licensed to industries, which are in the phases of trials. One product already in the market, and many such there which we are doing, animal trials, as well as, we aspire to go to the humans for its application. Now, I'll take you through, few specific examples, and I'm not going into the technical jargons. So this is one example where we have reconstructed, created meniscus. Now, meniscus is what? In the long bones, we have a soft tissue which allows cushioning of the body so that the bones do not collide, and it balances the body, and it allows us to do mobility. Because of injury, because of trauma, sports person, old age people, osteoarthritis, this gets herniated and it has to be removed by surgery. So doctors would do is suture it and try to repair it. Many times it is beyond repair, so they have to scoop it out and remove. And they replace this with a plastic material which is available in the market. Now imagine a human living tissue is replaced with something which is plastic, but there is no solution because that's what is available. Right, so we thought, can we make a humanized version of this meniscus, because there is a huge demand and there is no such donor because nobody has an extra pair of meniscus to give. So, and it's a huge market that is there. So we started working on the design concepts. What we have understood from the native meniscus, we have mimicked down to 1 micron dimension in the multi-layer structure, and this is exactly what it looks like, and one can create this less than 15 minutes taking coordinates from a real patient. What we did next was to prove that this meniscus is able to, be biologically active. What we did, we took stem cells. Stem cells are what? These are special cells we have in our body, which are progenitor cells, and is the ability to differentiate to any kind of tissues in the body. So we grow these stem cells, then populate them in the meniscus, mature them, culture them in a bioreactor, give a lot of shear forces, which I was saying. Finally, after a couple of weeks' time, we can see they attaining the strength that's like a native meniscus because it should not fail. Then we attained the extracellular matrices like the similar native meniscus, and they are ready for transplanted. We have transplanted this to the animals and we have shown that they are compliant, but we are now going for more experiments in large group of animals where we want to test it. Next interventions come where in the meniscus itself there is minor injuries, traumas where it has to be replaced. So we have developed an injectable gel. So instead of the entire meniscus being replaced, people are looking for minimally invasive technologies, meaning if I am hurt, I do not want to visit the doctor and the doctor performing a surgery on me, because that is always expensive. It has its own downside, also health concerns. So if the treatment can be done with a minimum surgery, minimal invasive surgery, then most people would like to go for it. So we thought, instead of an open surgery in the earlier case, can we repair it through a simple injection at the site where the injector would load polymers which are loaded with stem cells, with biologic factors and growth factors. So we have proven with our studies, in vitro and in the animals, that once it is put at the site, it naturally forms a bridge, repairs that. There is modulation happening in the meniscus. In a couple of days, weeks time, it gets fully matured and we can get a repaired meniscus. This is something, a very, very interesting and very, very important medical intervention, what the doctors are looking for, this kind of meniscus surgery. Next, what we did was, we further improvised this and made it patient compliant, meaning and less expensive. See, any technology can be very expensive, but it may not reach to everybody. So as a researcher, as scientists, our job is to bring technologies to every individual in terms of affordability, in terms of compliance, which is acceptability. What we have done here is, we have taken patients' own blood cells, we have removed the platelets, and activated, we call it PRP, plasma rich platelet, that have a lot of growth factors. This, when combined with the polymer, when injected, we need not to add any growth factors which is external, cutting down the cost, making it patient compliance at autologous system. It is not rejected by the body, and it can do better regeneration as we have shown in the meniscus of this tissue. Next example is for a bone regeneration. What we have done here is we have created special bone forming materials which we have 3D printed. Now bone, as I was saying, when there is a bone repair, the patient cannot come back to work immediately because there is a period of rehab where the forces, slowly the patient is made to exercise so that the mineralization happens and the repair happens, and after a few weeks of this rehab, the patient can go to regular activity. So this advantage of this bone repair scaffolds which we have done is they carry some actuation molecules which, when introduced into the bone, when the patient is transplanted with this bone material, one can, through a magnet, actuate this, when the patient is in the bed. So the repair and regeneration of the bone is much faster, because what we are doing externally through the magnet is exercising those bone cells, and because of these forces, external forces, bone cells are secreting more extracellular matrices, mineralizing more minerals and becoming stronger. So the rehab required period is much lesser, and we are creating a much ambient environment there itself through mechanotransduction. There is a lot of science that goes into it. I'm not going into that part, but now we have a technology. Instead of a simple ceramic material, we have a activated material which can form bone in a much faster manner. The next study, what I'm showing to you, is about repairing cardiac ailments, which is the heart. Heart is a tissue, an organ which its job is to pump blood throughout the body, and again so that it reaches nook and corner, and all the tissues gets oxygen and nutrition. Now couple of things happen because of which the patient may have to visit the doctor, and it can be fatal as well. One is myocardial infarction, which is nothing but the heart attack. Next, babies are born with hole in the heart. Now current intervention says that stem cell therapy, where stem cells can be injected at the site, because this ischemia, because of lack of oxygen, the cardiac tissues die out, because of which the cardiac cells are not able to beat properly, they leading to a secondary comorbidities, and it can be fatal as well. So what is the solution for this? The solution is, if you can create a patch. So we all know about Band-Aid, but this is a Band-Aid which is made out of cardiac cells, meaning heart cells, and it has a special property of beating itself similar to our heartbeat. Sounds easy, but it's very complicated. But again, how we did was, we took stem cells, we differentiated them to cardiomyocytes in the 3D printed structure. We matured them in a bioreactor for the forces, because cardiomyocytes need actuation, the force. Also it need electrical signals to mature. Once done, what we have done, we have included blood vessels. If you look at, our body has a lot of blood vessels which supply nutrition and allow us to exchange waste. So the red that you see in the top are blood vessels which are created by bioprinting endothelial cells, and the green lines that you see are the cardiomyocytes. So finally we have a construct which are alternating layer of blood vessels and cardiomyocytes, and what we have shown here is this patch, what we have created is a stamp size patch. It's a living structure and it beats. So it's a very unique structure, and it beats by itself, and we can keep it beating. So we have got 73-75 beats per minute, which is very similar to the human heart rate, right? Now how it can be used? So it can be used in patients where there is a myocardial infarction. The doctor can take it and put it with a glue or stitch it at the site, and it would help the heart to revive and function with the normal beating. Second, babies who are born with a hole in the heart, this patch can be put at the site and stitched so that this leakage of blood in the different chambers can be prevented. So it has huge healthcare requirements. We are now doing animal experiments for this because any technology that you develop has to go through animal experimentation, large animal experimentation, then clinical trials, human trials, then come to the market. So this is so all the technologies, whatever I've shown, each one of the slide took us 5 to 6 years to reach that stage, though I'm probably saying in couple of minutes, but there's a lot of back-end science, a lot of technical understanding, research goes into creating this. Now I'll take you, I understand this is a pharma department and a pharma conference, so all these are all medical related, but this is probably very interesting to you because as a pharma department or a school, when you are developing any molecule for a potential treatment, you have to validate it in the cells, then you validate it in the animals, then you validate it in the humans through trials, then it comes to the market. And this is all about 12 to 15 years or maybe sometimes longer. Many times, most times rather, even after validating them and successful trials, it has to be withdrawn from the market, right? And it has to be done, tested in the animals. Now, ethical concerns of killing animals is very, very strict and it is very, very expensive, So, it's a very long procedure, very expensive procedure. People have thought out, including us, we are working to creating an alternate to testing these molecules in the laboratory. Meaning, instead of injecting them in the animals and testing them, can we create liver, for example, or a disease model in the petri dish where you are emulating that disease and the molecules can be tested. So, it will be an intermediate between cell lines and an animal study so that you can screen a lot of molecules very, very fast. So this is about creating in vitro liver in the petri dish where drug toxicity can be, because one of the most important questions that you would like to ask for your molecule is whether it creates efficacy, whether it creates toxicity or not, right? And that is done by injecting into the animals, then you look at the liver profile and functions and that you derive. If you can do everything without putting in the animal and not killing an animal, in a very precise manner, that is what we are doing. And it is more so important because globally animal testing is being banned, or there is a thought that in the times to come it will be completely banned. If the animal testing is banned, where do we go? So these kind of models have come up a long way, and the government is also pushing globally to adopt and make these kind of models. So what we have done here, so I'll not go into the technical jargons, but what we have understood is in a liver, one unit of the liver is the acinus. We have a parenchymal, non-parenchymal cells. We have stellate cells, hepatic cells. We have so many other Kupffer cells, so many other cells which do a function. So what we did was we deconstruct. So we are not making the entire liver. We are making a lobe of the liver like a Lego toy. And if you want more drugs to be testified, you can put more of these models together as one unit, and you can do a test. So what we have done, we have taken stem cells and hepatic cells which were differentiated from stem cells, stellate cells, Kupffer cells, and 3D printed them hierarchically so that they behave and they are structured similar to what we have in the liver, right? Having done that, so at the top you see these triangles with yellow and the pink. These are the two kinds of parenchymal and non-parenchymal cells structured similarly as we have in the liver so as to achieve a function. So then what we did was we looked at the hepatic functions, because I can claim it to be a liver, but it cannot be a liver if it doesn't show the hepatic functions. What the liver hepatic functions are? Those who are working in this, it should secrete albumin, it should secrete urea, it should secrete LDH, CPR activity. So we have all cross-validated that, and we have cross-validated with the animal results available. We are very, very excited to say these simple models actually can emulate like a liver, and they behave like normal liver, what we have in the body. So once that is done, what we have done is to challenge them with drugs. now to understand whether they can, give us, outputs, readouts of hepatotoxic drugs, non-hepatotoxic drugs, and idiosyncratic drugs. So why these three categories? Because if I have a liver model, if I am challenging with a hepatotoxic drug, my liver cells will die with time. I'll get readouts similar to what is happening in an animal model. If it is non-toxic drugs, the liver cells will remain the same and they will not die out, and we'll get a idiosyncratic drugs, meaning they behave very abruptly, and which is very difficult to predict in animal models. We were challenging with that. So once we did, I'm not going into the, the bars and the graphs, but it tells us very precisely that the liver cells able to metabolize these drugs and give us readouts very, very similar in pattern to what it happens in the animal system. And that is very, very exciting to us, because now we have a model which we can now further work on, and make it a product for selling to pharma companies. But you cannot just sell this like that, because it's a very complicated setup. So what we did was, we miniaturized this and made it into a cassette. So imagine we will be selling a small cassette where it will have liver growing inside. It can be a cirrhotic liver, it can be a fatty liver, it can be a normal liver, it can be a liver infected with malaria, liver infected with hepatitis A, B, and C. So that's what our portfolio would be in the future. So why? Because the pharma company who was developing a molecule against that can immediately test on it. But we also want this to be easy for the pharma companies. So what we have done, you will appreciate here. So what we have used, we have microfluidics. We have miniaturized this into a very small cassette. We have understood the flow parameters, we have understood the nutritional requirement. So it has now two chambers, one is inlet and one is outlet. The pharma companies would just inject the drug, it will go to the liver chamber, the liver grown here would metabolize this, then the readout would come, they would put into spectrophotometer or any other assay, and then would want immediately they can get a readout, and we can make this replicative, even thousands, 10,000, million times. And that is the beauty of this, because this is done in 3D printed, 3D bioprinted. So one can recapitulate, because this is one very important parameter, because batch to batch variation is something pharma companies and in medical research is not acceptable. So you have to produce something which behaves the same time and again. Because of its automation, we could achieve that to an extent, and we are further working on that. We are now collaborating with groups for making a malarial version, Plasmodium vivax, so that anti-malarial drugs. Now we are working with ILBS Liver Institute so as to make hepatitis A, B, and C versions, so that we have more versions of that. Then these are some more studies where we have shown that compared to the dynamic system, compared to the static system, dynamic system behaves better. Dynamic means we introduced a flow channel. So instead of, rather than a static condition, our body is not static. Blood is flowing through the organs, and there is a constant supply of nutrition and taking out of waste. So we have created that through perfusion system, through peristaltic pump, so that we match that flow of blood, as in the liver, and we are doing it, and we have got much better results with that. So this is the timeline. Again, we have tested this, as I said, non-hepatotoxic, hepatotoxic, idiosyncratic, many drugs we have tested. We have looked at genomics, proteomics studies to confirm that what we are claiming is true, and filed a patent as well for this technology. Next, I think the last one is we are also working on making skin models. This is something very important, because if you look at the global scenario, skin models, animal testing of cosmeceutics and pharmaceutics on skin is completely banned. So it has to be relied on in vitro models. Currently, few companies from America and Europe are doing business globally, and there is a huge scope of revenue as well as resource generation in that. So understanding skin, what we have created, again through 3D bioprinting, we have created skin on inserts which are on petri dishes, and we could now do a lot of testing. So we have done dermis, epidermis, we have the barrier membranes. So what we can look at through these models, we can exactly predict sensitivity, irritation test we can do. We can say if a company selling cosmetics says that when you apply on your skin, you will, the skin tone would go down, meaning you will be more fairer or something like that. It cannot be tested in, humans or in animals. You can put it here. We have melanocytes growing here, and because if it is a positive effect, you can see the effect there. You can test anti-wrinkle activity. We can test permeation activities. We can do a whole lot of things, with the skin models that we have. So these are some of the studies that we have done. So finally, we have this model which we want to translate through a startup company and to take it to the market. Also, we are taking this skin and making bigger grafts for third degree burn injury patients, because these grafts are currently bought from Western countries. Also, there is a great demand, and most times these patients are relying on these grafts, which are pretty expensive. So we have developed these skin grafts which are transplantable for patients who are suffering from third degree burn injury. Also, as I said, we look at different parameters to validate our hypothesis. This is some studies which shows the irritations test, and some of the other tests that I claimed. So with this, I'll close, but because I can see a lot of youngsters here. My summary and request would be for you to, whenever, because you are at an age where you are trying to understand which direction, which topic, which research area, which industry, which job to look for, right? So try to question yourself what excites you more, right? We questioned ourselves, we understood that, there is a huge requirement for this. We have a training in that. We collaborate, and collaboration is very important. We do collaborate with pharmaceutical companies. We collaborate with a lot of labs across the globe, to bring out what we are getting it out. Thanks to the media houses who have highlighted the outcomes, some of the technologies which have been released by our health minister into the market. And the most important slide. So all I have shown, this is my picture of my lab group members, and thanks to all of them for putting up an effort, all the funding agencies and the collaborations across the globe, and thank you so very much for your time.
Congratulations, Dr. Mondal, for your nice work and explaining this to us very nicely.
Thank you.
I know that you have tried to bring down this to our layman's level, because the scientific intricacies would be much more complex for us to understand. So, if there are any questions.
Thank you, sir, for showcasing your research. It's a very interesting topic. I would like to ask you one question. What about the aging and multiplication of these tissue cells?
So, in the body, we do age, right? Any tissue that you look at, it replenishes itself with time, and there are progenitor cells around which kind of replaces that with the aging cells. So are these models. So if it is an in vitro model, we do not worry much, because the time of screening is 20-25 days. After that, it has to be thrown out, and new batch would come. So it has nothing to do with the aging. If you look at the organs and the tissues, right? So if you have a kidney or a liver, right, which we have maybe transplanted or the bone, life inside and the cells are very smart, right? They have a turnover time, right? When it is integrated, they know, the niche environment knows how to put the cells in a quiescent environment. Not all the cells are in active state, right? But of course they have a shelf life. When they die out, new cells, progenitor cells, that's why we have a reservoir of stem cells, and they again differentiate to occupy those missing cells, and that is how the cycle goes on. So if it is a liver patch or a cardiac patch, new cells from periphery would be coming into that space, because it is accessible both through the angiogenesis, which is the blood vessels, right? They can move around, and the scaffolds are so designed that they would have spaces, and interconnectivity for the migrating cells to come in. So it's a self-sustaining system. It's not an isolated system. So as the heart is, we do not replace the heart, neither we bother, what the cells has happened to them. So same way, the system, takes care of it. That's the beauty part of it. Because the cells are very smart. All you have to do is to put the right cells at the right location, and put it through a logical meaning through the surgery at the site. That's all. They take care of it.
Once again, very much thank you, sir.
Thank you.
Any other question? I would like to ask one question. Do you take the patient's cell all the time to start with, or you take from other donors also? And if you take from donors, what about the genomic composition?
Yeah, thank you, sir, for the question. So it's a very potent question from the perspective of translation, because doing science in the laboratory is a different thing. But if we want to translate, then it has to be answered. So if you look at the current regulatory approvals, the hurdle is the immune rejection, right? Because of the cross genetics. So ideally, if you want easier regulation and to bring this to the market, autologous cells are the best. Meaning, if I'm making a meniscus or an intervertebral disc or a liver or something, I can get the patient's bone marrow or the stem cells or fat derived. We work on adipose derived stem cells. We work on iPSCs, somatic cells which can be made into embryonic-like stem cells and iPSCs. So being autologous, it is not rejected by the body, right? But there can be situations where the patients are aged, right? And they do not have the right quality of stem cells, because they will also be in a depleting number as well as the condition. In that case, iPSCs is one good change that has happened. One can create from skin cells to stem cells, right? But that is also a lot of regulatory hurdles are there, because it's a very new concept, right? And there is question of tumorigenicity and many other things. In that case, donor is another alternative. Like we take donor HLA matching. So that is where from the nearby relative or who has a genetic makeup similar, donor tissues are taken and from their cells are taken. But ideally it should be the autologous. That's what, so that all these complications are avoided, and it is very much doable, because adipose derived, for example, bone marrow, earlier bone marrow was the source of stem cells or blood stem cells. Now fat derived stem cells have come in, and they can be differentiated to many kind of cells. Yeah.
If there are no more questions, then we can thank Dr. Mandal with a big round of applause.
Thank you so much, Dr. Biman B. Mandal, sir, for your thought-provoking presentation. Your work has provided us with a deeper understanding of advanced tissue engineering and its application. And also thank you so much, Dr. Mahendra Kumar Modi, sir, for chairing this session. Now, I would like to request Dr. Manish Gautam, sir, Associate Professor, Faculty of Pharmaceutical Science, Assam Downtown University, to felicitate Dr. Biman B. Mandal, sir. আৰু এটা ধ্ৰুপজ্যোতি ছাৰ আছেনে এতিয়া ধ্ৰুপজ্যোতি ছাৰ আছেনে নাই Now, I would like to request Dr. Dhrubajyoti Sarkar sir, Associate Professor, Faculty of Pharmaceutical Science, Assam Downtown University, to felicitate Dr. Mahendra Kumar Modi sir. Now, I would like to request Dr. Farooq Alam sir to come over and felicitate Dr. Manas Patim Sarma sir, Dean, Faculty of Science, who has evaluated this session. A short announcement. Now we will be continuing with the next session, and lunch will be organized after the completion of this session. A short announcement. Now we will be continuing with the next session, and lunch will be conducted after the completion of this session. I repeat it again. Now we will be continuing with the next session, and lunch will be organized after the completion of this session. I request all the delegates not to leave the hall.
Good afternoon, everyone. I am Drishti Sen, your host for this session. Welcomes you all to the last scientific session of AdtU Pharmacon 3.0, where we continue to explore research and transformative advancement in pharmaceutical science. This session, like the others, resonates with the central theme of our conference, empowering healthcare and well-being towards achieving sustainable development goals, research, innovation, and industrial academy partnership. To preside over this session, we are privileged to have Dr. Suresh Deka sir as the chairperson. Dr. Suresh Deka is a distinguished microbiologist with expertise in bioremediation, biosurfactants, and plant microbe interactions. He holds a PhD in microbial ecology from Guwahati University and completed his postdoctoral research in biosurfactant at University of Ulster, Northern Ireland, UK. With over 3 decades, the academia and research experience, Dr. Deka has served in various capacities at IASST Guwahati and is currently an advisor at Assam Downtown University. His prophylactic research output includes numerous high-impact publications and several patents related to biosurfactant production and applications. Dr. Deka has successfully led multiple extramural research projects sponsored by DBT and DST, focusing on bioremediation, phytoremediation, and biosurfactant applications. He is a PhD supervisor and an active member of esteemed academic societies. Recognized with prestigious awards and fellowship, Dr. Deka also presented his research internationally, contributing significantly to the advancement of microbial biotechnology and environmental sustainability. We are honored to have you with us, sir, today. It is now my privilege to introduce our eminent speaker for this session, Dr. Kushagra Khanna, sir. Dr. Kushagra Khanna is an accomplished researcher and academician with extensive contribution in the field of pharmaceutical formulation development, specializing in nanodrug delivery and radiation science. Dr. Khanna is currently serving as the head of research and assistant professor at Faculty of Pharmaceutical Science, UCSI University, Malaysia, where he also coordinates QS ranking and industrial collaborations. He holds a PhD from Delhi Pharmaceutical Science and Research Institute, Delhi, in collaboration with INMAS, DRDO, and has been actively involved in international and national research collaborations. Dr. Khanna has authored over 40 international reviewed research papers, contributed to 8 book chapters of artificial intelligence in drug discovery, and holds 2 granted patents with 1 patent under filing. His expertise in chemical, biological, and radiological disaster management has led him to train numerous defense personnel over the past 7 years. Dr. Khanna's work has earned him numerous accolades, including the Best Emerging Researcher of the Year 2023 by UCSI University and Outstanding Service Record 2022 by Laureate Group of Institutions. He is also an inducted member of esteemed organizations such as Royal Society of Chemistry, UK, the American Chemical Society, UK, the Indian Association for Radiation Protection, and the Indian Pharmaceutical Association. Dr. Khanna has delivered invited talks to various international conferences across Indonesia, Italy, and Thailand, and his research in nuclear medicine has targeted drug delivery, continues to make a significant impact globally. I now invite Dr. Suresh Dekar, sir, to kindly welcome our esteemed speaker and take over the proceedings of this session.
Hello. Thank you for giving me such opportunity to share this session. And I am also a host, actually. I am working here as an advisor. So thank you, the organizer, and very nice introducing. I accept the invitation and I now proceed for the session. Now, I request Dr. Khanna. Your title is Khanna? Yes. Though you are from not our India at present, but I think you are the origin of India. Okay. So, I request to deliver your talk.
Okay. So a very good afternoon, everyone. And I would like to thank, first of all, the organizing committee and also the AdtU for inviting us and to share our basic research where we have been, spent more than a decade to serve basically for the nation as well, because earlier, before going to abroad, I was with DRDO for almost 8 years. So from there, we started to use a new technology which we would like to be, shared with everyone throughout the global, right? So today, my overall talk will revolve around a term called nuclear medicine, when we comes to nuclear medicine, the first word that we hear is nuclear. We immediately goes to bomb, nuclear bomb. But apart from that, the nuclear has its lot of advantages when we look at from drug delivery point of view. We as a pharmacist should be knowing that how we can serve the population with more effectiveness and to reduce the toxicity. Correct? So this was first of its kind where we started in India from Defense Institute itself, where we use nuclear medicine, a radiation source, to evaluate all the delivery systems. You talk about any delivery system, you can evaluate without doing anything, just an image, and you can predict each and everything from that image. Whether you want to know the distribution, you can. If you want to know the pharmacokinetics, you can. Even if you want to know where it has been targeted, yes, it can be done. So my whole talk will be sharing some of the areas where we have found extensive results and where it became a groundbreaking innovations there, right? So first of all, the principle and example of pharmacoscintigraphy, we called it pharmacoscintigraphy. It is a coined term by us itself because we are using pharmacy and using a nuclear medicine which is established one of its kind as in the medical field. So we use pharma with scintigraphy, we termed it as pharmacoscintigraphy. So in this, what we do, we use nuclear, or we say we use radiation as a source to evaluate all your delivery systems, right? So all of us knows we have oral, we have inhalation, we have dermal, right? We have injectables, we have so many delivery systems. Now we have nano going around, correct? But thing is, how to achieve or how to know whether your delivery system you have developed is better than others or not, right? We have to wait for animal studies. We have to sacrifice so many animals to get one product out of it. Then we have to go for human trials. It takes a lot of years, lot of efforts, lot of cost. But when we introduce this technology, we are actually cutting the total tenure of development, because you get the result on the same day within an hour. The interpretation is within half an hour, within an hour, you get all your results in your hand, and then if you need, you can go back, troubleshoot your formulation, come back and do it again. It's always a very precise way of doing your things in developmental process, right? So the first is your scintigraphy, meaning it's a non-invasive imaging technique. You don't need to get blood taken out, you don't need to sacrifice, you don't need to put any efforts where the invasive technique is used. It's a totally non-invasive technique where just a simple image captured under the camera will give you all the information about your product, or we say a formulation. So it employs radio-labeled drugs to track their behavior. Now what happens is, if you have a drug, you can bound it to a radioactive material. The most common radioactive material that we use is your technetium. Technetium pertechnetate, we use it as one of its very common radioactive source. Now this has a half-life of 6 hours only. So once you deliver the drug into the body, within the 6 hours it will automatically come out. So it's totally non-hazardous for the body. Because we have been using technetium, we have been using iodine, thallium, gallium, all you are using in nuclear medicine from past so many decades, and it has found to be safer. The dose that doctors give in your X-ray, the X-ray that you take has more exposure to your body than this. It is 75% lesser when you go for this type of studies, right? When you go for X-ray, it is more than what this dose gives you, because X-ray is what? Radiation again, right? Here also radiation is there, but it is 75% lesser than what you get in your X-ray, a simple X-ray. So this, what we do, we have a molecule, we bind it to a radioactive compound like technetium, which is very commonly used because of its half-life, right? So when you administer this compound into the body, wherever your compound will travel because of your formulation nature or physicochemical characteristic, your tracer molecule will go along with it. If it has to deposit in the stomach or it has to declare a mucoadhesive nature, it goes and stick to the stomach lining, and your radioactive moiety will keep on showing you in the camera till it detaches itself from the membrane, right? So after going into the body, at least you know where my molecule went. It was to be targeted for mucoadhesion. Did it really did mucoadhesion? Or was it just taken and excreted out from the body? So it becomes one of the area where you can use your label compound to track throughout the body where your molecule is traveling. Now, why is it important? Now, important comes from ADME. From this alone images, we can find out your absorption, distribution, metabolism going in the body, even excretion. So when excretions comes or absorptions come, at least we are very much able to recognize where our molecule went, where from which organ it started its absorption, in what time it came into a systemic circulation. So you get a very handy type of data within minutes or hours that you can correlate with your findings or with your innovations. Next is your radiopharmaceuticals. Now, radiopharmaceuticals is a branch where if you go to a nuclear medicine center, radiopharmacists are required. I don't know how many of you know that this is also a career option doing a radiopharmacy. We have been doing pharmacist, but radiopharmacist is also a career option where you will find a lot of people who are trained to elude, to develop a radioactive pharma compound, right? So all those nuclear medicine centers have radiopharmacists which are liable to detect or to evaluate the dose to be given to each and every human, right? So what type of common radio tracers we are using is, we are using technetium 99 MTC, technetium pertechnetate we called it. We called it iodine 131, radioactive iodine, and we have fluorine 18, which we use in PET imaging, right? Positron emission tomography. So now we comes, why nuclear imaging is required? Why actually we need to go for all this, where we have to expose ourselves to radiation, where others have to be exposed to radiation? When it comes to exposure, the exposure is very minimal. That exposure is being protected by a lead shield behind which you will start working. So for a person who is dealing with radioactive isotopes, is completely safe. But for the patient who is being given the drug, or we say for any human trials where you're using the radioactive compound or tagged compound with your formulation, for them it is safer in terms of X-ray, right? So all those, cancer patients are undergoing radiation therapy. That radiation is quite high and have a lot of side effects. But when you are evaluating anything, or when in nuclear medicine, if we are giving any of the, you have renal scans, your kidney scans are being conducted very oftenly, your lungs scan are being conducted for COPD patient very oftenly in nuclear medicine. These are some of the area where it is established. You have nothing to question to that accuracy there. Now, as per the latest update, US FDA has approved the data produced by gamma camera or scintigraphy for the approval from US FDA. So this is the latest they have announced that every data that they get from scintigraphy can be used for approval of the drugs. Now we see why nuclear. First is radio tracer mapping done by following injection. We have been seeing, we know that all IV injection directly goes to the systemic circulation. We all know this. But have you ever thought the permeability or penetration of your subcutaneous or intramuscular, how slow they can go? So this can be easily detected by using imaging there. Then you have distribution of activity. After going into the body, you take any formulation, oral, dermal, inhalation, any. After it goes into the body, how it travels into the body, how much time it takes from coming out from the stomach, how much time it takes to reach to that target body, it's been calculated automatically by the system. You have the image in your hand, you can clearly see the anatomy, you can clearly see where it has gone, in what time it has traveled, from which destination to which destination. So it becomes a mapping of your product or formulation there. Then you have radionuclide produced gamma rays. Now we cannot use any material to tag a molecule and put it into a gamma camera. Your molecule should be emitting radiation. It should be a gamma radiation, more specifically if you have to use that material to bind towards your drug, right? Then detection system produces image. So once you administer any radioisotope tagged with your molecule into a human or animal, you will get an image that we will be seeing in the coming slides how the images looks like, and you can predict from that images how your formulation performed inside the body. Was it successful or was it failure? Because we have been doing for a lot of companies these things, and we have actually challenged them that it failed, right? Then you have computer creates data. So everything, your manual involvement is very limited. You have actual real-time images going in front of you, and from that you can easily track back and easily translate what you have been doing in your past formulation. So it's very easy to troubleshoot your formulation or troubleshoot your experimental in your wet lab. Then medical information generated. Now suppose we are using an inhalation product to be tested. That means we need to see how much of the drug it got deposited in the lungs after a inhalation. If a patient doesn't know he was a COPD patient, in case he was a mild COPD patient, he was not knowing this, and he actually volunteered to be a patient for this particular test, this can give him a picture that you are in a mild COPD case. So that images will show that there is some obstruction in the lungs which can be modified or which can be now to be cured first before you move on to a severe conditions. So medical information is also generated parallelly when you do all these types of tests. Next is where you can use this scintigraphy now. For drug evaluation, number one, new application filing, therapeutic drug monitoring, and drug formulation development. Now when we look at all these four, we are actually making them tested from phase 0 to phase 2 trials. Phase 0 is proof of concept, right? So if you have developed any concept, it can be proved either on one animal, two animal, just to make that yes, it works actually, right? So you need not to sacrifice and just waste so many animals, because now we are moving towards artificial intelligence, we are moving towards a lot of new generation equipments where we need to minimize the wastages. Now this is how the machine looks like. You might have seen in a lot of hospitals this type of machine. This is called as gamma camera. Now gamma camera, when you look at, it has two heads. Now this is your first head, and the second head is at the bottom of your bed. Now these two heads actually captures and image either animal or human. Now this will only be able to image if that body is containing a radioisotope bound molecule. The molecule can be your formulation, the molecule can be your drug, right? It can be anything, maybe a food also. Because we did an experiment with Maggi. We tried to radio label Maggi, and we have done to evaluate GERD, gastroesophageal reflux, right? So you can make it for anything. The only science is how you label, how you make sure that your radioisotope doesn't leave your molecule when it goes into the body, right? So first thing is your radio labeling of drug or molecule, then radio labeling of formulation or excipients, and then you have a third way of interventional nuclear medicine. By this way also, we can do some of the experiments that we will just see. One or two I have put for you there. Now potentials of pharmacoscintigraphy when we comes is first is drug evaluation. Now this type of the methods or experiments gives you the drug release itself also. You don't need to get into the dissolution apparatus or there. One time when you are performing an experiment, you will get all data parallelly every time. Correct? So then you can have in vitro animal biodistribution, phase 1 trial for human biodistribution, 2 and 3 phase for efficacy and evidence-based medicine. And another is phase 4, where we called it as post-marketing surveillance, PMS, where a lot of industry comes because they have already launched the product into the market. They now need to undergo PMS to maintain their product marketing, right? So even phase 4 goes for this type of studies. Now what are the priorities in pharmaceutical research? We have to develop a new drug, a new chemical moiety, modification for better efficacy, like all what we are doing in pharmaceutics. Whosoever making formulation is trying to make it more effective, trying to make it more efficacy with reducing the side effects or toxicity. Then evaluation of pharmacological potential of herbal products, then novel drug delivery system. So these are few of the priorities where pharmaceutical research nowadays is making its way. Now first we see the things which we can do actually. First you can screen your drug molecule if you have to evaluate the properties. If you have, before going to a formulation, if you have to see the nature of your drug, you can do that with scintigraphy. Another is under pharmacology, you can do dynamics, you can do kinetics, efficacy, pharmacological action and toxicity profile, right? So these are very small area where we focuses while during the drug development or formulation development part. Now what are the prerequisites for radioisotope? What makes a molecule a radioisotope? Number one, it should not affect the nativeness of your drug. That means it should not change when it's bound to a molecule. It should not change its physical and chemical properties. If it is changing its physical chemical properties or any other properties, even if it's changing the efficacy, it's not a radioisotope. Then it should have a suitable half-life. We always try to take those products which have a shorter half-life so that it is easy to excrete out from the body. Then you have radionuclide should emit gamma rays or X-rays and not beta. Then you have a low radiation dose, of course, and in vivo stability. In vivo stability means when it goes into the body, it should be into a intact form. The breakage of the radioisotope should not happen with the drug molecule. If the splitting of the molecule and radioisotope happen, it will show you some other images giving you a false data. Correct? So it should not be broken when it goes inside the body. So few of the, we are using technetium, 90% cases we use technetium, then you have thallium, FDG for PET scan, then indium, gallium and others for cancer oncology purposes. So some of the application, very small applications that we can use scintigraphy is gastro-retentive formulations, right? Then you have mucoadhesive systems, oral dosage form, and also your inhalation formulation where you check the lung deposition and clearance from the lungs. So all those can be evaluated single-handedly with a single image of any one volunteer or animals. Now another thing is a case of a drug called nalbuphine. Nalbuphine is a known opioid, synthetic opioid of morphine category, right? So what was done was, we all know that it's a painkiller and it has a ceiling effect. Morphine, in case of morphine, as you increase the dose, your respiratory depression increases. But in case of nalbuphine, it becomes ceiling. After a certain dose, it doesn't show any respiratory depression. So this was the case when we used for defense person. Whenever there is a blast in the forces, or there is a gunshot, very combat casualties happen, and there is a mass casualty basically, we need to have one of its preparation that is easily accessible to all the army persons or defense persons. Because when we see the defense personnel areas, all those are hardly graduates, right? Those are your first responders there on your field. So to make, in case of mass casualty, what is the alternative system apart from IV? IV is the first that we always choose to relieve from pain, right? So, but in case of battlefield scenarios, we didn't have IV as option. Reason was, you have a mass casualty. Number of medic and paramedic staff in all the defense forces are very limited. The time when a medical person reaches to the other patient, it has already went into some serious trauma, right? So we need to have an alternative system. This was a challenge given to us to come up with a way to have a better efficacy like IV. So what we did, now this is an image. If you see, on your left hand side is a rabbit, right hand side, both are rabbit images. In rabbit, we gave the injection from your chin. We gave it from your chin. And it acts so fast that you can see the body of the rabbit from the first second frame itself. If you see the first and second frame, your body outline of rabbit is very clear. But if you go to your right hand side, that was given intramuscular, the injection. Same drug given intramuscular and the same drug given from the chin, we called it submental area. Now, submental area very easily accessible. Even it's not covered. Defense forces have helmets and all those things. These is not much covered, so it can be easily given. Moreover, no need to find vein here. Just take the injection and put it inside, right? In case of IV, you have to find out the vein and chances that you can inject somewhere else also. So this case, when we did on animal, it showed very promising result with nalbuphine as its kind, because within few seconds only, it showed the whole animal structure. And same way, when we were calculating the, you see the graph at the bottom, when we were calculating the, elimination, the first graph is for submental. You see, the elimination is going very fast. The graph is declining very fast, but in case on the right, it is very slow. The graph is quite slow. So you can predict the release and the distribution kinetics of your drug from single image here, right? Now, same we did for humans. In humans also, we give it in the submental area and we give it in the arm, your muscle, deltoid. Then what we found is, if you see in your left side images, you will see the heart clearly visible, right? It means the drug immediately went into the systemic circulation. That's why your whole body outline is easy to see. But in case of intramuscular there in your hand, the first few 4 or 5 images are quite blur. There is no outline except your bright spot. That bright spot is nothing but the area where you injected your injection. Correct? So it became a bright spot. After that only you can see the outline of your body images. Correct? So it became one of the alternative way of administering a drug if you need a faster action. But it was done for defense person. Now next was your floating tablet. We have always, read about floating tablets. We have already read about mucoadhesive tablets. So floating tablets, if you need to evaluate whether it floats, because outside in the beaker when we do experiments, yes, it floats. Yes, it releases the drug, what we want. But when it goes into the body, half of the drug doesn't float. So we did on human and we found that this in stomach, the tablet is still floating there. Right? And it's slowly releasing all the content out from the body. So what you do, whenever you have to draw, whenever you have to see how much drug is there, just draw a region of interest. We called it a sphere, circle, like we have done here. This gives you a count. Your initial count was 5 and your remaining count is 2. That means 3 has already been absorbed there, right? So you can easily do things when you know your pre-dose that you administer and your final dose. So it is easy for you to calculate between both of these doses. Now next was for colon. When we did for colon, because colon is the last part of our body, if a drug has to release in the colon, it has to pass all your pH, it has passed all your enzymes, right? Gastric pH. Then only it can reach to colon. By this time, half of the drug is released. Now this was a case that was given by a company. They didn't tell us that the tablet formulation is failing. They only said that we are getting complaint from the doctors and patients that the drug is not effective. They only said this one. So what we did, we tried on 5-6 volunteers, given them with the radio label tablet, their marketed product. We took from the market, labeled it, we gave to the volunteers, and when we see few volunteers only get drug release in the colon. Out of 6, 3 volunteers showed no colon release. Only 2 were showing colon release. This we shared result with the industry, and they then told us the actual story, saying that we were getting complaint that tablet is coming out from the body intact without getting disintegrated, right? So they have to go back to the lab. They took whole batch out from the market, recalled the batch, again did and introduced the batch into the market. So that was a PMS study, post-marketing surveillance. So because colon, very difficult to find out whether your drug reaches there or not, right? You are administering from the mouth, reaching the last part of your body, detection is quite difficult. So we can be doing with this technology there. Next is your interventional nuclear medicine. So many of you have been listening about nanoparticles, solid lipid nanoparticles, lipid nanocarriers, right? So what we did an interventional study with a radioisotope called radiothallium. Radiothallium absorbs in the body very normally, right? Like a normal molecule. But what we did, we gave a radiothallium to the animal. After that, what we did, we gave a lipid nanoparticle, LNP. Now what we saw was, lipid nanoparticle slowed down the absorption of radiothallium in the body. So what make us think is, if you have any radiothallium contamination in future, if you have a thallium in the body, if you have to stop the absorption of the thallium, give some lipid so that the release or the absorption of your thallium goes down, right? So it became a new intervention. Nothing, it was going a regular check. We were giving thallium for some study, but after that we gave nanoparticles, lipid nanoparticles, and it shows the images of declining the absorption. Next was the case for iron. lot of people take iron supplements, right? Lot of people, are being prescribed on iron supplements for their bone health. So we also did the same thing. We wanted to check whether iron get absorbed or not. So what we did, we gave iron to the animal, right? And what we did, after giving iron, we injected a radioisotope called technetium MDP. That is a bone seeking agent. It only goes to the bone. So if your iron is not absorbed there on the bone, those area will not show dark. They will be lighter. That means the absorption was not there prominent. So we evaluated from the image itself by creating the region of interest, how much was absorbed, because we know what we gave technetium MDP initially and how much it has reached to the bone. So you can easily calculate your percentages there. So even we can use for FMCG, for detergents also we have done, for finding out which of the detergent or soap basically clears your stain, because we radio labeled the dust, we have radio labeled the particles there, put it on the cloth, and then we have been doing all this activity to find out which of them has a better efficacy. So we could not publish because it became a conflict of interest later that we have defamed any of the brands available for detergent. Now this is a case for pulmonary, where on your left you have the patient with COPD. We try to give salbutamol. That was our first drug under this scan. We give salbutamol, radioisotope, tagged it with the technetium, gave it to the human through MDI, and we can evaluate the images how much obstruction is there in his lung. So you can see on your left there are obstructions there, and on your right is the healthy lung, right? So you can easily find out how much obstruction is there in the lungs for your pulmonary. If the patient complains that he's not having a good breathing there, or if he tired, is very fasting, so you can evaluate there. The last case was for salbutamol topical cream. Another thing when we were working there was in high, in your Siachen level, high altitude area where you have very cold temperature, you have frost bites there. Your hands, your feet often go frostbite, right? So what we did was we took salbutamol, which is a vasodilator basically, right? We developed a cream and we did on hands. We first did on hands. So when it's a frostbite, your circulation is not enough in your hands. Your fingers are not visible there, right? But after applying salbutamol cream, the next image you see, your hands, fingers were quite visible. So we evaluated cream with this type of technique, and we evaluated that salbutamol not only for inhalation but can be used for dermal delivery as well. Function is same, dilation, correct? The next and the most emerging area after this is your theranostics, where a lot of you have been hearing about, precision medicine, theranostics, correct? Now theranostics, we will not go in much detail because it's another science, but it's a part of scintigraphy basically, where you have a molecule, you make it radioisotope, but it's in a way that it is for diagnosis, it's for imaging, and it's for treatment. So if all three things are being conducted, it becomes a theranostics. You have giving a therapy there. So for cancer patients, we have been giving this for more than a decade nowadays. So the first example for this is your monoclonal antibodies, right? Here you have examples of ibrutinib, and then you have lutetium-177 labeled PSMA. So these are few of the drug that has been turned out from its original way into a radio-labeled molecule given to patients for treatment, diagnosis, and also for your imaging purposes. Next, we talk about emerging trends in scintigraphy. So the emerging is we are developing new tracer molecules throughout so that we can have a PET and SPECT tracer molecules now, right? Then you have a target-specific radio-labeled drugs that comes under theranostics, that particular molecule is tagged with a radioisotope that only goes to that organ without going anywhere, if it is tagged properly. Like I shared one example of MDP. MDP is a bone-seeking, right? So it only goes to bone, it doesn't go to any other organ there. Then you have hybrid imaging modalities where you have been able to combine SPECT CT with PET imaging. So you have a 3D structure coming out when you are doing any study. So evaluation becomes easy. Your target, your organs where you have been seeing is easy, because sometimes we gets in a confusion whether it becomes a stomach, it passes to the duodenum or ileum, because very close organs there in animal cases. Then we have nanotechnology and theranostics being developed nowadays, lipid-based and polymeric nanoparticles for their stability. Then you have AI-driven algorithms, personalized medicine and pharmacokinetics modeling, real-time distribution and kinetic modeling. So these all are areas where you can do it with a simple molecule, take, label it, put it inside the body, and you can study whatever you want. Your final output will always be from the image. If you know the image, if you know the anatomy, you will get your results out in an hour. We don't have to wait for HPLC to develop method. You don't have to wait for any other high-end equipment. So you can do it in an hour or so. Now future advancement is like, first is integration of AI and machine learning for data analysis. Still we are using, nuclear physicians to interpret the results, which take a lot of time, because in India or in anywhere, the load of patients in nuclear medicine is very high. People go for renal scan, people go for thyroid scan, people go for lung scan. All these are very common nowadays. So they have a lot of load. Now we are trying to, integrate AI so that AI can receive the image, interpret, give it back. So it saves time. Only the nuclear medicine expert has to see whether it is being translated correctly or not, right? Then you have expansion in applications where you are making a tailored treatments for patients. Like in precision medicine, the concept is tailoring the treatment. It's not one treatment for all. It is a treatment based on the patients now, right? So this is another area of expansion for applications. Now, what are the beneficial for academic and pharma industries? Drug delivery study. How this scintigraphy can help in pharma and academic is to study the drug delivery, kinetics, formulation development, drug retention, distribution, biodistribution, targeted delivery, and proof of concept of the formulation. So anything, if you have in your mind, you can think of labeling and putting it inside. It has no side effects, it has no adverse effect. The dose is in microcurie, 100 microcurie, and that 100 microcurie is nothing. And it is getting out from the body 6 hours. The half-life is 6 hours, right? So it has nothing to do with toxicity there. The final and the conclusion for this is that pharmacoscintigraphy, a powerful tool in drug development, formulation optimization, and theranostics. So all of us as a pharmaceutics or formulation development people don't have to wait for days and months to get one optimized formulation, because that formulation has to go to analytical, HPLC method has to be developed, then release has to be seen. All those big steps can be cut down, make it to one day. You prepare formulation, test it, not doing well, troubleshoot it immediately, do it next time, right? So it becomes a very easy process for the drug development or formulation optimization. Then real-time tracking. nobody can challenge your results. What is being displayed in the picture is the real time. Whatever, you cannot manipulate the picture. It's a real time. It's a radioisotope being given, and it shows the actual image, what it happens in your body. So you cannot manipulate the image. So it becomes a real time evidence there. Then you have integration with AI learning, and novel radio tracers is expanding in application of precision medicine now. And finally, the challenge is the cost and expertise. We have very few, a handful of people in India or globally which can do this process and which can actually conduct all the developmental process in one single image. Right? So expertise is needed. Yes, cost is involved, of course, but it's not too costly that cannot be bared by anyone. Right? Another one is that continued research and collaboration between academia and pharmaceutical industry will drive innovations in drug discovery and development. So all the academic people, we in the society, are basically to help or to come out with innovation that can resolve the problems in the society, or we can do a transfer of technology to the industry to come up to make it available for all the public. Right? So with this, I'll end my lecture for this. Thank you very much.
Thank you. Thank you so much, Dr. Khanna, for your insightful and thought-provoking presentation. Your work has provided us with a deeper understanding of healthcare policies and pharmaceutical education. Now we have a brief question-answer session. I invite the audience to ask their questions.
First, I'd like to offer my thanks. Nice talk, very nice talk, and important topic. So, audience, all are tired, no, all are hungry, but you can ask any few questions regarding the nuclear medicine, regarding his talk or any related to his talk. Audience can ask. Any questions? No questions? Any? So if no question, actually I have several questions in my mind when he delivered his lecture, because I am a layman, because this is not my subject. But due to constraint of time, only few questions, that is layman question, you can say. That is first question regarding the nanomedicine and your nuclear medicine. What are the difference? Nanomedicine and nuclear medicine, both are the emerging technology, thrust area of research.
Okay. Thank you very much, sir, for your question. The nuclear medicine, the basic difference, if I revolve around, is nuclear medicine develops or works around a radioisotope. Nano will not work with radioisotope. Nano formulation or nano medicine is decreasing the size to the nano based, right? But when we talk about nuclear medicine will go around the use of radioisotope in any medicament to treat the patient.
And another I think in nuclear medicine, it is not invasive, but nanomedicine, I think.
Yes, sir. If you talk about nuclear medicine, it's a process there. So it's a non-invasive technique. Nanomedicine has nothing to do with technique. It's the type of carrier system there, right? But here we are talking about a technique to evaluate your nanoparticles, nanomedicine. Anything you can evaluate with this technique. Even if you can do, the labeling of biscuits. I just have a very small experiments. I did a project, atta and maida biscuit. Which of them goes faster gastric emptying? Was very, a thing which lot of people know that maida retains more there or atta retains more there. So it becomes a very small topic. Just label, eat maida biscuit or atta biscuit and see your gastric emptying time there. So it becomes very easy and handy for us to do.
So, actually this is the area of research, but I am asking regarding toxicity, regarding the, I know the X-ray, if you use the cell, will damage. But here also, it is the same thing, I think.
Sir, that dose is 75% lesser than what you get in X-ray. So it is already being there as nuclear medicine is being treated, your technetium. It is 6 hours is half-life, and the dose is 75% lesser than what you are getting in one exposure of X-ray. So it's completely safe.
It's completely safe.
It's completely safe. Because otherwise, FDA would not have approved this technology to be used for drug evaluations.
I have seen in your slide regarding the radio, what is term radionuclear should not eliminate beta. Why?
Sir, because gamma radiations are easy to capture, right? Beta very difficult to handle also. So we always take all those radioisotopes which are gamma emitting. We don't go for beta. We cannot capture them. Gamma can be done. Gamma, even at the, how to say, the decomposition process is not very easy for all those things. It has to go in a lead shield chamber. It has to be burrowed inside a bunker there. Beta cannot be contained in that also.
OS regarding radionuclear OS, where we will dispose, because it will again react in the environment.
Sir, we are disposing. If you remember, there was one incident in Delhi itself. One of the Delhi University professor, without knowing, he just put that radio emitting substance into a bin. And it created a lot of havoc. It created a radiation in whole Delhi area, Narayana area, that it was called, and a strict action was taken against that professor. The waste of radiation is not to be put like normal waste. It has to be put into a lead shield first. It has to be given a time so that all its activity goes zero. Then only you can treat it as a usual waste. Because technetium, 6 hours, half-life is coming. We hardly use 500 microcurie. So in 6 hours, 250 microcurie, another 6 hours, 125, 72 like that. Within 24 hours, you will get certain microcurie which will never harm anyone. So it's easy to dispose there. But when you go for iodine, thallium, their half-life is 265 days. Right? So you have to be very careful while you are making them into a waste there. So we use very limited things for this.
Thank you, Dr. Khanna. Your nice presentation in the, actually this third area of research. So I congratulate, and please give him big hands for research.
Hello, sir. Sir, you have mentioned one thing, salbutamol, that is you are conducting a study to give a topical. So my two questions. First question, it will be giving as like a transdermal patch, or we will be giving cream? Okay, cream solution will give. So sir, as it is targeting as a beta-2 agonist, so beta-2, so it will be mainly targeting the beta-2 cells, and that is receptors that will be present. Mainly we are focusing on nowadays bronchodilators, as well as beta-2 are presenting on the heart, as well as mucous gland also. That is you are targeting through topical route. So is there will be any effect on the heart or the lungs when we are giving topical solution?
Okay, a good thing, a very scientific one. See here, when we talk about heart or there, on those, you are going for systemic circulation, correct? The dose. Here we are using only 1/4 of the dose, 25% of the dose, so that it just dilate, blood flows in. We are not treating like what we used to do inhalation basis. We are just making a dose that only dilate your veins, blood flows in. That's it. So for making that, it is not salbutamol alone. As a formulation, you have to put penetration enhancers, you have to put a lot of excipients there to make it go deeper and do its action there.
Okay, sir. Thank you, sir.
Now, I would like to request Dr. Saikat Sen sir, Dean of Faculty of Pharmaceutical Science, Assam Downtown University, to felicitate Dr. Khanna sir with a token of appreciation. Now I request Dr. Dhrubajyoti Sarkar sir, Associate Professor, Faculty of Pharmaceutical Science, Assam Downtown University, to felicitate Dr. Suresh Deka sir. As we conclude this session, I would like to express my sincere gratitude to Dr. Khanna sir for sharing his expertise and invaluable insight with us today. I'd also like to extend my heartfelt thanks to Dr. Suresh Deka sir for chairing this session. Now, I request all the delegates to proceed towards the K Block cafeteria for the lunch. We'll be continuing our session after the lunch at 3 p.m.